It's great to be here. Thanks very much for the invitation. The talk that I want to give you today is mostly going to look at the evidence for evolution that we see in a number of different areas of science. Last night we talked a little bit about my journey, how I had started by rejecting evolution and then later came to accept it. So before we get into it, I just want to ask one or two questions along this line. Is science an acceptable activity for a Christian to partake in? Sometimes in our culture, there's this idea, a common idea among folks who aren't that familiar with science. They think that science and Christianity are opposed to one another, that they're competing with one another in some way. Historically, that's actually something that has not been a part of the Christian tradition over time. It's actually fairly easy to make a case that Christianity was a major influence in how science came to be, and that the reason why people started doing scientific activity was because they were convinced that nature should have a rational basis because it had a creator behind it. So we can ask this question: is science an acceptable activity for a Christian to engage in? Here's a quote from Reformed theologian Abraham Kuyper, where he says very clearly, there's nothing within creation that does not belong to God, and as a result, it's a very Christian activity to explore and understand that creation with the tools that God has given us. Here's another quote from Sir Isaac Newton. This is what Newton had to say. Newton, of course, in his day, was at times accused of sort of removing God from the equation by showing how certain physical phenomena could be explained by laws. And his thoughts on the matter was that understanding the sort of law-governed cosmos was cause for rejoicing that God had put these laws into the cosmos. So he didn't see these things as in conflict. Here's a statement from Trinity Western, my institution, and we have a statement on creation, and it frames this issue quite well, I think, and so I like to quote this, where we emphasize at Trinity that we accept two sources of revelation, that God has revealed himself to us not only in the scriptures, but also in the created order, and that these are both forms of revelation that God has given to us. And as a result of that, science can be an act of worship where we investigate the properties that God has built into his created order. Another way to put this is something that I mentioned last night, the idea that if both Scripture and nature are forms of God's revelation to us, that they can inherently have no conflict between them. Now, we appropriate special revelation and general revelation with different tools. Special revelation, we use exegesis and hermeneutics to understand the revelation that God has given to us in the Scriptures, and God has given us the scientific method. I'm convinced that the scientific method, or using our logical faculties in this way, is a God-given activity to understand his created order. Now, the challenge might be that we might think that there might be an apparent conflict between what we're seeing exegetically and what we're seeing scientifically. But it's my conviction and the conviction of many other people that fundamentally there isn't a conflict, and that these two forms of appropriation of these different sorts of revelation can actually benefit one another. We can actually use exegesis and hermeneutics to perhaps rein in some excesses, philosophical or theological excesses of science. So science might say things along the lines of, well, humans don't have a purpose, or, the created order of the cosmos is just the result of an accident or something along those lines. And as Christians, we can say theologically, well, no, there's things that theology and philosophy can say to science to sort of rein in its excesses where it goes beyond what it's able to say. But on the same hand, we can look at science, and we can use science, if we're doing it correctly. Science has the opportunity to speak to our exegesis and hermeneutics as well. There's been examples in the history of Christianity and science where this process has taken place as well. So say for 1,600 years, up until the time of Copernicus and Galileo, say, the church was convinced that exegetically and hermeneutically that we lived in a geocentric universe where the earth was the static center of the universe. Now, based on the science and the convincing evidences that came to light at that time and moving forward, the church realized that it had to go back to the exegetical side of the equation to say, we've been thinking about this scripturally for 1,600 years along these lines, but that doesn't seem to fit with what we're seeing in the science. So that prompted the church to go back to the exegetical side of the equation to say, maybe we've missed something. Maybe we've been inappropriately taking our culture and imposing it on the text as opposed to letting the text speak for itself. So it may well be in the case of evolution that we're at a similar juncture, where what happens on the science side of the equation may cause us to look again at fresh eyes with the text, not because we're going to let science dictate what theology should say, but because these are both forms of God's revelation. If we're seeing something on the science side of the equation, it could well prompt us to look again at the text with fresh eyes and to try to take it on its own terms. Okay, so a brief outline of where we're going to go today. We're going to briefly talk about science as a way of knowing. Then we'll talk about evolution as a scientific theory, some of the evidences for evolution, specifically some of the evidences also for human evolution. So, and then at the end we'll maybe comment a little bit again on the sort of two forms of revelation model and offer some concluding thoughts. Okay, so let's talk a little bit about science and how science works and how it is a way of knowing. So when you hear the term science in common usage, I'm sure your professors have hammered this into you, that when you hear someone say, oh, well, that's your theory, well, Joe's theory is that, this is what's going on. That's a very different usage than what we see in science. In science, theory has a specific meaning, that it's an explanatory framework that's withstood repeated experimentation, makes all sorts of predictions that can be tested, and as it's something that we have not yet been able to falsify through the scientific method. So in colloquial usage, as we mentioned, theory is something more akin to a guess or a conjecture. This is actually closer to the scientific term hypothesis. So if you say something is only a theory, it's actually quite a high compliment from a scientific viewpoint. A theory is something that has withstood repeated experimentation, and we have not yet falsified it through the scientific method. So it's not to say that it's absolute truth, because certainly new evidence may come to light that will cause us to modify our ideas slightly, but it's the best idea that we have at the time about the way things work. So various theories, there are various theories in science. As a geneticist, I speak frequently about the chromosomal theory of inheritance as one theory that's quite important in genetics. If you're a microbiologist, germ theory of disease is quite an important theory. And one of the things that we'll go on to talk about here in a minute is that evolution is actually the most foundational theory that there is within biology. I sometimes joke with my physicist colleagues because they know that they don't yet have a grand theory that unifies a number of different areas that they are working with. And I say, yeah, biology's had its grand theory since the 1800s. We've had, when evolution came along. And they say, yeah, but it's all fuzzy and doesn't make all, so then we have nice back and forth. It's whether or not physics is the better science or biology is the better science, right? Okay, so moving on to evolution as a scientific theory, one way to put this is, despite what you may have been told as a Christian, evolution is a theory in the scientific sense, not in the colloquial sense. I first saw this cartoon at a talk that Francis Collins gave, and I thought it was clever enough to grab and bring along. So even goldfish crackers seem to have this challenge. So we can say that evolution is a well-tested explanatory framework. It's supported by a large body of experimental evidence. It makes accurate predictions, and we have not yet falsified it. Now, we've changed it significantly since Darwin's time. Darwin, for example, had no idea about how genetics works. Excuse me. And as a geneticist, I just want to take the poor guy out for coffee and explain to him how it all came out later, because there's this big chunk in the middle of Origin of Species where he's just completely muddled about how genetics works. We have evidence that Mendel was aware of Darwin, but we don't have any evidence that Darwin was aware of Mendel, and it wasn't appreciated. Mendel's work wasn't appreciated until the early 1900s. So we've certainly shifted some ideas, but Darwin's core ideas about how evolution works, that idea we have not yet falsified despite 150 years of effort. So there's a very famous quote by a Orthodox Christian population geneticist where he says, "Nothing in biology makes sense except in the light of evolution." Okay, so if I tell you that evolution is a scientific theory, that means that there should be a huge amount of evidence to support it, and there certainly is. So we'll now take a brief tour through some of those lines of evidence. We'll start with biogeography and then look at these other areas in their turn. So biogeography is just simply looking where species are on the planet and musing about how they may have come to be there. It was biogeographical evidence that first sort of twigged Darwin and also Wallace to this idea that perhaps species weren't fixed and that they shift over time. So one example would be that oceanic islands lack endemic terrestrial mammals. So an oceanic island is an island that's not connected to a continental landmass. They're volcanic in origin. Hawaii would be an excellent example. And what Darwin noticed is that when he visited these various oceanic islands, that they didn't have any terrestrial mammals that humans hadn't introduced. They didn't have their own endemic mammals. And Darwin mused on this, and he thought, well, why is it the case that the Creator didn't choose to make mammals for these islands? Because the territory was, or the environment was very suitable for mammals, and it was the case when mammals were introduced that they thrived. So Darwin was musing, why aren't there terrestrial mammals? Now, there were aerial mammals. These different islands often had their own endemic bat species that were not found anywhere else, but they didn't have terrestrial islands. And the other thing he noticed that whenever he saw the species on a given island, that they seemed to be most closely related to the species that were on the continent, the nearest continental landmass. So for example, when he's on the Galapagos, he notes that the birds and the species that he sees on the Galapagos have the unmistakable stamp of the American continent. And he's previously been to an archipelago off the coast of Africa, and he notes there that the species there have the decided stamp of being African species. Yet the two island groups that he's looking at are actually very, very similar to one another in their climate and their topography, and yet the species that are there seem to be most closely related to what's on the nearest continent. So this got him thinking, and these observations, among others, eventually led Darwin to propose a hypothesis that species share common ancestors. So the idea that he came up with would be that populations would somehow become genetically isolated from one another. They'd stop swapping alleles back and forth. This would mean that genetic changes that are happening through mutation and recombination in these different populations would no longer be averaged across the population as a whole because they're now separate, they're isolated from one another. So this means that as differences accrue and accumulate, that they wouldn't be shared between those two groups anymore. So average characteristics of those populations may begin to shift away from one another. So this might lead to new species over time. One way I like to sort of use, or one example or an analogy that I like to use to explain this process is something that's a little bit more familiar to us, and that's how languages shift over time. So speciation is a lot like how languages shift over time. You can have populations of speakers of a common language that are separated, language changes that accumulate over time, syntax, spelling, new words perhaps. They're no longer shared between those two populations. If you have these two populations that are separated long enough, that the average characteristics may shift because of those differences in those different populations. And this may lead to new languages over time. So we can think of both species and languages as information systems that are copied imperfectly. And because of that imperfect copying, there's this possibility for average characteristics of a population to shift over time. Okay, here's just one example. I wrote this up for Biologos a while back as a way to sort of illustrate this process. This is probably not, unless we happen to have somebody who's like an Old English scholar in the room, this is not likely going to be something that's an easy read for us, despite the fact that this is English, well, Anglo-Saxon. This is John 1:29 from the West Saxon Gospels, about a little over 1,000 years ago. It's not really comprehensible to us, Here's the Wycliffe edition from the late 1300s, which is a little bit more familiar to us. Here's Tyndale in the 1500s. Here's the original King James. So this is the 1611 edition of the King James. The King James that you're probably most familiar with is a later edition, the 1800, the Cambridge editions from the 1800s. And then if we look at a modern translation, something like the NIV, it reads very naturally to us. Now the point here is that these populations, this is a continuous population of speakers. Nobody sat down one day, in 1394 and said, well, we've been saying it like they have been in 990 uniformly up until this point, but it's time to shift. Let's get everybody together and decide how we're going to speak the language differently now. This is a continuous population where average characteristics within the population are slowly shifting over time. And as a result, we can see the progression leading to what we see in the present day. So in a sense, this is a little bit like a fossil record, right? These are samples at discrete points of what, in fact, is a continuous process over time. And it's a continuous process within a population. How do, languages shift over time incrementally? Well, they start with new ways of speaking being introduced into the population by one or two speakers. And then those ways of speaking might become more common over time. And then eventually they might end up taking over the population such that everybody thinks that that's the right way to do it. But it doesn't happen suddenly. It happens incrementally over time. They have this process of incremental change because there's this imperfect copying mechanism. And species are very much the same way. New mutations can come into a population. Those mutations can become more common over time, over generations within a population. And because of that, average characteristics of the population can shift. So if you have populations that are separated, those processes are now uncoupled in the two populations, and that change over time can slowly, very slowly, lead to new species, things that are different from one another. One example I don't have here that I could show you is how English is related to other languages. And if you speak more than one language, you probably know about many correspondences between English and the second language that you have, especially if it's a Romance language. And the evidence is that those languages, if you go far enough back in time, that they share a common ancestral language that's been shifting slowly in separate populations over time. Okay, so the process is a gradient over time, whether in species or in languages, because it's average characteristics of a group that's shifting over time. Okay, so let's look a little bit at some evidence from paleontology. So because evolution is a scientific theory, it continues to generate new hypotheses. One hypothesis that evolution generates, and this was actually even known at the time of Darwin, is whales, the group of organisms called cetaceans, whales and porpoises and dolphins and things like that, because they're mammals, the evidence would be, or that evolution would predict, based on their mammalian characteristics, that they are actually the descendants of four-limbed terrestrial mammals. If you think about mammals, mammals are tetrapods. They have four appendages. We're mammals, we're tetrapods. Whales were kind of the odd organisms out because they just have front flippers and a tail. They don't have four limbs. So Darwin actually proposed, he sort of hypothesized in the first edition of Origin, that maybe whales were descendants of bear-like creatures. He had seen some evidence from somebody in North America who had observed black bears swimming and catching insects in the water. And he thought, okay, maybe that was sort of maybe how this got started. And he was absolutely ridiculed for it. I could show you quotes from Christian apologists just slamming him because it's such a ridiculous idea that whales would be the descendants of these four-limbed terrestrial organisms. So that's this hypothesis. It makes predictions about what we should find. And one of the things that it makes a prediction of is that the fossil record, if indeed there was this transition from four-limbed terrestrial organisms to whale-like creatures over time, we should be able to see species in the fossil record that give us a sense of that transition. Now, it's not likely that we're actually going to find the actual ancestral lineage that leads to present-day whales. The probability of that is very small that we would actually find that. but it should allow us to see what sort of species were around in the fossil, using the fossil record at what different time periods. So even if we can't find the actual lineage, we should be able to find their relatives, and their relatives could be informative as to what sort of characteristics they may have had. And one of the things that this Christian apologist who slams Darwin, he says, look, the fossil record is clear. There's nothing that blurs the distinction between whales and bears, right? You've got four-limbed organisms, you've got whales, and there's nothing in between, because nothing was known at that time. It actually took paleontologists a long time, actually, to finally figure out where to look for these sorts of organisms, because it wasn't clear where on the planet this transition had taken place. It turns out that places like India, Pakistan, and whatnot are the place that you need to look. And one of the reasons why it was a bit delayed is because it's a great area to do paleontology, but it's not a great area because of the political instability in part of the region. But once it was worked out that this was the place to look, a lot of examples that blur the distinction between these different species were found. So what we see in the fossil record, we see modern whales up at the top, and we have other species in the fossil record that, based on their skeletal morphology and some of the particular characteristics of their skulls, we're quite convinced are related to whales. That some of these species, such as Pakicetus and whatnot, they have certain skull features that, up until the point of their discovery, were thought to be unique to whales. And as you can see, the different sorts of species that we've found greatly blur the distinction between present-day whales and four-limbed organisms. We see aquatic organisms with four limbs, and we see species that have greatly reduced hind limbs, like, say, the basilosaurids there, sort of midway up on the left. These are species that are fully aquatic, but yet have very small hind limbs. So we haven't yet rejected the hypothesis that whales are the descendants of terrestrial four-limbed organisms based on what we see in the fossil record. And again, we don't say, well, that proves it. We say we haven't yet rejected that hypothesis. Embryology is another area for whales that has some interesting insights for us. One of the things that you could predict based on this observation is that if you look at the embryology of whales, that they might retain some features that are reminiscent of their evolutionary trajectory. Now, it's not an absolute guarantee because some of these features could have been lost, but it's often the case that when you look at the embryological development of organisms, they tend to have features that are reminiscent of their evolutionary trajectory. One of the reasons for that is it's a lot easier for evolution to sort of work around things instead of sort of redesigning things from scratch. So one example would be in present-day cetaceans. So these are dolphins. They actually do have four limbs. So they are good, true tetrapods in that sense. But they only have four limbs for a very short time of their development. So like all mammalian embryos, when they start off early on in development, they make forelimb buds that will eventually become the forelimbs. And they also make hind limb buds at the proper time that any tetrapod embryo should be making hind limb buds. And you can see them here as well. So the way this actually works is the developmental program that says make a forelimb bud and make a hind limb bud that runs normally like it does in any other tetrapods. But what happens in the hind limb region a little bit later on is a second developmental program kicks in that basically says, no, don't do that, stop, and pulls it all back in. So that's a good example of sort of evolution finding a workaround. As a tetrapod, you start the hind limb just like any tetrapod should, but then later on, something else gets cobbled on top of that that says, no, pull that back in. We don't need to make a hind limb. Forelimbs are good enough for us. Another example is how nostrils form. Cetaceans don't have nostrils on the front of their face like most mammals do. Mammals typically have those two nostrils on the front of the face. As you know, dolphins and whales have a blowhole on the back of their head. What we see in dolphin embryos is that they too, like any good mammal, start off with two nostrils on the front of the face, as you would expect. And then over the course of their development, those nostrils that started on the front of the face migrate developmentally to the top of the head and fuse to form a blowhole. So again, it's a great example of a workaround. You start with the general structure, and then you can imagine over time, in the lineage that led to modern-day whales, that we had modification to shift the blowhole farther and farther back. And actually, I don't have time to deal with it now, but if we went back and looked at those different species in the fossil record, we see examples where the nostrils progressively are moving back in those different species, as well as species that have smaller and smaller hind limbs. So embryologically, we see these features in whales that match up very nicely with what we would predict based on an evolutionary trajectory leading to them. Okay, that's all well and good, but I'm a geneticist, and I'd like to think about genetics more than morphology and paleontology and whatnot. I'll say this: if we had not thought of the idea of evolution up until this point, when we started sequencing genomes of organisms, it would have forced us to this hypothesis simply based on the sorts of things we see when we compare the genomes of organisms to each other. So one of the predictions in the case of whales is that their DNA might retain features that are best explained as having once been adaptations for a terrestrial environment. So the idea is we can look in the DNA of modern-day whales and see if there are any things left over that suggest a terrestrial lifestyle in the past. The reason for this is the enzymes that do DNA copying, they don't know per se what they're copying. Their job, is just to simply copy the DNA sequences as carefully as they possibly can. So what can happen is a gene can pick up a mutation, and even after that mutation happens that might render a gene non-functional, the enzymes doing the DNA copying don't know that that gene sequence is non-functional. So they'll attempt to copy it as faithfully as they possibly can, even though it's no longer doing its function. So in this way, you can have genes that are mutated that are nonetheless transmitted for a large length of time because of that copying process. So these are just some papers that illustrate this idea. Say the short wavelength sensitive cone pigments. These pigments are very, very useful for seeing through air. Now, whales don't typically use their eyes for hunting in the same way that other mammals do. Whales have shifted over to using echolocation because they hunt in a very low light environment. But they still have the genes for the pigments like any good mammal should have for seeing in not in a low light environment. Here's another example, olfactory receptor genes. So these are proteins that are expressed in the nasal epithelium. They bind onto air-based odorants, and that binding changes the conformation of those proteins. And that change in conformation leads to things that eventually are interpreted as changes in nervous system activity in the brain, and we use that as a sense of smell. So these are for air-based odorants. Now, whales, especially this is now in odontocete whales, so these are toothed whales. Toothed whales seem to have completely lost their sense of smell. They don't even have an olfactory organ on their blowhole anymore that we can determine. But just like any good mammal, they still have, mammals have about 1,000 genes devoted to olfactory receptors because the sense of smell is incredibly important for mammals, for most mammals. But here's a lineage for which the sense of smell is now completely, dispensable. They still have the genes. The genes are there, but they're riddled with mutations. So whales, toothed whales, even though they don't use a sense of smell for hunting anymore, they still have all the genes that you would expect for an air-based hunting organism, except those genes have now been inactivated due to mutation. So what we end up with is this sort of scenario where we have converging lines of evidence from these different areas of biology that continue to support this hypothesis that whales are the descendants of terrestrial four-limbed organisms. So comparative anatomy and physiology, the fact that whales are mammals was the first line of evidence. Skeletal morphology would be part of this as well. We've seen in the fossil record and embryology and with DNA sequencing, we have all these different lines of evidence that converge on this same conclusion that whales are the descendants of terrestrial tetrapods. So we would say we have not yet been able to reject this hypothesis. And this is now something that's beyond a hypothesis. This is now something that scientists accept as a lineage. that this was the transition that took place, even if we don't yet have the full details of all the steps along the way. So it's a good example of evolution as a theory, working in that theory sense, where you have broad explanatory power and you have evidence from a lot of different areas. Okay, you might not lose too much sleep over the question of whether or not whales evolved. Typically, the question that Christians are more concerned about is, okay, well, what about us? Did humans evolve, right? Okay, maybe whales, maybe that's okay for whales, but is it okay for us? And these lines of evidence, similar lines of evidence, exist for human evolution as well. We're not going to hit all of them. We'll talk a little bit about paleontology, we'll look at some genetics, and then I'll briefly talk about some population genetics as well, because this pertains to the Adam and Eve question. Okay, so is there any evidence from paleontology? Are there any lines of evidence that, or any species in the fossil record that blur the distinction between what we would consider human and other forms of life on the planet? So even back at the time of Darwin, Darwin proposed that humans would share common ancestors with other great apes, things like chimpanzees and gorillas and orangutans and such. And the reason for that is just the similarities in skeletal morphology. Even Linnaeus, when he did his classification back in the 1600s, he put humans in with those other species. And the reason why they're called primates is because of prima, the Latin prima, which is sort of first. These are the top of the ladder, as it were. So even, you can't accuse Linnaeus of being a Darwinist, but he put those species together as well. So the short answer is yes, there are a large number of species in the fossil record that do blur the distinction between what we would consider human and what we would consider ape-like organisms. These are species that are known as hominins. Hominins are any species that are more closely related to humans than to chimpanzees. So these are any species, after we part ways with chimpanzees, any species that are more closely related to us as opposed to the chimpanzee lineage. Humans and chimps share a common ancestor at about 4 to 6 million years ago, so these are species that are since that time. And again, the point is that we might not be able to determine the exact trajectory that leads to our lineage, but these are species in the fossil record that are very likely relatives, at least, of that precise lineage. And the place to look is in Africa. Darwin predicted that Africa would be the place, and indeed, it ended up being the place. So a lot of hominin fossils have features that are intermediate between what we see in apes and what we see in humans. So here's one very famous species. This is Lucy, Australopithecus afarensis. This species has adaptations for walking, so they're bipedal, but they also have adaptations, skeletal adaptations for climbing. There are other species in the fossil record. So Homo erectus is a species that is very similar to us morphologically from the chin down. The early ideas about how evolution worked were, the thought was that it was all about changes in brain, so the idea in brain structure. So back in Darwin's time, scientists thought that what they should be looking for is an organism that has a very ape-like body and a human-like brain. As it turned out, that's actually completely backwards. The story of human evolution is pretty much that our postcranial skeleton, everything from the chin down, evolved first, and then later on we added more and more brain power to that basic body form. So if a Homo erectus, sidled up to you at the bus stop today, you would notice that they're quite different in the fact that they have very small cranial capacity. They have a much smaller skull and brain size than we do. But everything from the chin down would be pretty much normal. We see other species as well. So here's Lucy, here's Australopithecus sediba. Again, not that the idea is not that these are necessarily direct ancestors of the human lineage, but that they're relatives, that they share very recent common ancestors with whatever that precise lineage was that leads to Homo sapiens. Okay, so here's Homo erectus, and we see adaptations for walking and for running. So at this point, we're looking at a skeleton that's very similar to the modern-day human skeleton, but with that reduced cranial capacity. And then the story of evolution from this point on in the human lineage is species that increasingly have a larger and larger brain capacity. And we start to see things like tool use and whatnot. So certainly when we look at the fossil record, we can't discount this idea that humans may have been the product of this evolutionary type of process. But like I said before, even if we had never come up with this idea prior, genetics and comparative genome sequencing would have forced us to this idea. So if we think about this again, we remember that evolution is a population-level phenomenon. It has to do with how populations shift their average characteristics over time. The basic idea is that a common ancestral population would have had a common genome. They would have shared a genome, which would have then had slight modifications after their point of separation. So when we look at the sequence, using modern comparative genomics, we can do a full genome sequence of the human genome, we can do a full genome sequence of the chimpanzee genome, the gorilla genome, the orangutan genome, and that sort of thing. Basically, what we observe when we compare those different genomes to one another is it very nicely fits with what evolution would have predicted. We see what are very small modifications between those different genomes, such that when we look at the human genome and the chimpanzee genome, they very much look like slightly modified versions of a common ancestral genome. So one example of this is when you do the actual raw sequence data and just compare at the DNA sequence level, the human and the chimpanzee genomes are about 95% identical when you put them side by side. And this is over about 3.1 to 3.2 billion DNA letters between the human and the chimpanzee genomes. When you line them up, they're 95% identical. So not only do we have the same genes as well as all the same intergenic sequences as well, we not only have the same sequences, we have them in basically the same order. So the idea here is that not only are the genes highly identical to one another, they're also in the same spatial pattern in the two different species as well. So like I said, our two genomes are exactly what one would predict as slightly modified versions of what was once an ancestral genome. Here's a figure from a paper that was done back in the 1980s. So this is 1980, so this is likely before many of us were around. But we've now, of course, done the sequencing to confirm the impression that this original paper gave. So what you see here on the left, well, with every pair, it's a comparison between a human chromosome and the comparative sequence in chimpanzees. And what you can see, just as you let your eyes sort of wander over with these different banding patterns, this is banding patterns that are just a dye that sticks to DNA, so it allows you to see sort of chromosome structure. What you notice is that there's a high amount of correspondence. There's some differences. The biggest difference is here. We can talk about that later if you're interested in that. But basically, as you look over the genome, it very much looks like a modified version of what was once an ancestral genome in one species. So that's sort of at the macro level. If you look at the level of individual genes, we end up seeing the same sorts of patterns. We made that point earlier about defective genes in whales. Well, those sorts of defective genes are also present in humans and in other species. And here's an example that we'll look at. Again, we'll look at olfactory receptor pseudogenes here in a second. So when a gene loses its function, even though it's still being copied, that gene now has a historical name of pseudogene. They were originally named pseudogenes because that means false genes. So when biologists found these defective genes in genomes, they still have most of the features of a gene, but they have mutations that show that that gene is no longer functional. Just like if you came across this as you were walking through a field, you would immediately know a few things about it. You would know that it once had a function, but that it no longer has that original function. The roof is missing, there's grass growing up through where the floor had been, that sort of thing. So it retains enough of its features to make you confident that it was a building, but it also has certain characteristics that make you confident that it no longer has its original function. And it's the same sort of thing that we see in genomes. We'll see the remnants of a gene, we'll see mutations that remove its function, but most of the gene structure might still be there. It'll still have the promoter, it'll still have portions of the coding sequence, that type of thing. So a biologist can look at that and say, yeah, that's a gene, or it was a gene, and originally they were called pseudogenes. It's just a historical name. So when we look at pseudogenes comparatively across genomes, here's the pattern that we see. So again, this is olfactory receptor pseudogenes. Primates have lost a lot of these as well. We don't have a very acute sense of smell because we're much more dependent on our vision for how we do things. So, and that's actually not such a bad thing. Can you imagine if you had a sense of smell that was acute as, say, the sense of smell that your dog has, right? We'd be like, somebody here had bacon for breakfast this morning. Somebody didn't have a shower, that kind of thing, right? So because we live in these groups, close social groups, it's probably not such a bad thing. But primates made a trade-off. We went with vision in ways that other organisms have. Like when I take my dog for a walk, he's experiencing the world mostly through smell. I'm experiencing the world mostly through my vision. So here's the thing. When we look at these olfactory receptor genes that humans have lost, so here's the human genome with these different human genes. These are all olfactory receptor genes. This is a paper that simply sequenced a number of them and asked what sort of mutations were present. And what we notice is there's many examples where we actually have identical mutations in identical pseudogenes between different species. So chimpanzee, gorilla, and orangutan. When you look at those mutations and the pattern that they make, they independently assemble a tree of relatedness that's identical to the other forms of evidence that we have for how these species might be related to one another. So we see two different categories. We see some mutations that are unique to a specific lineage, and then we see other examples of mutations that are shared. So these three are identical between humans and chimpanzees. These three are shared between humans, chimps, and gorillas, and these 6 are shared between all 4. Now, there's 2 hypotheses how you could explain this pattern. The one hypothesis is that these species are not related to one another, and that these mutations happen to independently, identical mutations happen to repeatedly crop up in unrelated lineages. So that's one hypothesis. The other hypothesis is that these mutations cropped up when these lineages had, or before these lineages had separated to become separate species. And if that's the case, then these mutations happen once within one population, and then as these other species separate from that population, they inherit the same mutation from that original common ancestral population. And obviously, the second example is a much more logical and much more parsimonious explanation than the first. The probability of having all of these mutations happen independently in unrelated lineages is extremely small, extremely, extremely small. Notice also what we don't see. For example, if we see a mutation that's shared by humans and gorillas, we can predict with great confidence that we will also see that mutation in the chimpanzee genome. If we see a mutation that's shared by orangutans and humans, we can predict with great confidence that it will also be present in the chimpanzee and the gorilla genomes. We don't see anything that's out of place that doesn't fit with this pattern. Okay. The fact that these sorts of pseudogenes can sit around in the lineage for a very long time also allows us to look for evidence for even deeper features that are from even deeper in an evolutionary trajectory. So one example that I'll talk you through now is the vitellogenin gene sequences and the vitellogenin pseudogene sequences that we see in placental mammals. So vitellogenin is a protein that is required for egg yolk formation. So amniotic organisms use, the ones that lay eggs, use vitellogenin as a way to put a whole bunch of nutrition into the egg yolk before the eggshell is deposited. Once the eggshell is laid down, then the mother no longer has the ability to supply the embryo with nutrition. Now, placental mammals don't have to do this because we maintain a placental connection with our embryos all the way through embryogenesis. So vitellogenins are these genes that are made in the liver of the mother. They're transported through the bloodstream. They're big, huge proteins, so they have lots of amino acids. Those amino acids are heavily glycosylated. They have sugar stuck all over them. And the three-dimensional structure of vitellogenins act as a lipid carrier as well. So it's kind of like this package deal, and you get to eat the wrapper too. So it's a fantastic way for egg-laying organisms to give their embryos a huge amount of nutrition before the eggshell is deposited. Now, placental mammals, such as humans and others, are, based on other lines of evidence, predicted to share a common ancestor with birds approximately 310 million years ago. I don't have all the time to sort of show you the lines of evidence that support that hypothesis. It has to do with skeletal morphology and whatnot. But the prediction is that humans should be descended from egg-laying organisms, but that we haven't shared a common ancestor with those organisms for a very long period of time. So this has got to be a paper that's the result of a couple of grad students sitting around in the lab one day saying, hey, I wonder if we could find evidence for vitellogenin genes in the human genome. Well, I don't know. It's been 300 million years. Do you think they're still sticking around? I don't know. Okay, don't tell the PI, but let's go look. Okay. I don't know. Maybe it was more planned out than that. So what this research group did is they said, well, let's look at the genomes of present-day amniotic organisms. So chicken as a good example, an egg-laying organism. Chickens have 3 vitellogenin genes, VIT1, VIT2, and VIT3. VIT2 and 3 sit side by side, and they're actually the result of a duplication in the past. So what this research group simply did is they said, okay, in the chicken genome, what genes are nearby the functional VIT genes? And they just noticed what those genes were. And then they said, okay, are those genes still present in the human genome? Yeah, those genes are present in the human genome as well, and they have the same sort of spatial pattern. So now you know what they're going to do next, right? They're just going to look very carefully at this sequence in the human genome and here to see if there's any correspondence with the vitellogenin sequences that we see in chickens. And when you do that, you find out that there is correspondence. So the black bars just indicate where there are sequence matches. And as you would expect, these functional genes have a high amount of sequence match. But we also see matches between the vitellogenin gene sequences and what's in the human genome, and also, I don't have time to show you, but a whole bunch of other placental mammal genomes as well. So there's fragmentary remains of sequences, vitellogenin gene sequences, in the human genome, precisely where evolution would predict that you should be able to find them. And as you might expect, because this is a very ancient sequence in the human genome, a lot of the mutations that remove the function of the vitellogenin gene sequences are absolutely shared between all placental mammals. It really looks like vitellogenin was lost in the placental mammal lineage long before placental mammals started speciating from one another. So here's just a small section of coding sequence from the chicken genome showing you the matches that we see with the genomes of various placental organisms. You can see here's a deletion in this sequence in the human genome. Here's another one here. And we can see that that's shared between a whole bunch of other placental mammals. So it seems like this sequence was lost before these different mammals went their separate ways. Okay, so those are all good examples of how information, different sequences might be lost over evolutionary time. I often get this question, well, okay, what about evolutionary gain? Is evolution capable of producing new stuff? Here's just one example that we'll walk through that shows how not only how mutations can remove information, but can also generate new information. This was a hypothesis that has been around for a very long period of time based on evidence that we see in present-day vertebrates. It looks like present-day vertebrates have doubled genomes relative to other organisms that are not vertebrates. And not only are they doubled, the hypothesis was that it was actually 2 rounds of genome duplication that sit at the base of vertebrate life. So what we see with the vertebrates as they diversify is that they might have had not just 1 round of genome duplication, but 2 whole rounds of genome duplication relative to other closely related organisms that aren't vertebrates. But there wasn't really a way to test this hypothesis until we could do large genome sequencing. So what we see here, okay, is the general idea of what is thought to be the relationship between various vertebrates. So tetrapods like us and fish are vertebrates. And then our closest non-vertebrate relatives. These are sea squirts, tunicates. So they don't look a lot like a chordate, but they are the closest relatives that are not vertebrates that was used in this study. So what was the hypothesis was long that there was these 2 rounds of genome duplication on this branch of the lineage that wasn't experienced by this branch. So the question then that you have to ask is how do you decide, or how do you identify which genes you might want to look at in present-day genomes to see if there was an actual duplication of the entire genome at a particular time? So what they were interested in doing, or let me just do this slide here for a second and then we'll jump back. The prediction is if you have these whole genome duplication events, it's going to produce, so here's sort of the ancestral genome and here's after the first duplication event, it's going to produce not just copies of every gene, but it's also going to produce copies that are in a particular spatial pattern. They're duplicates, but they're duplicates with a duplicated pattern. So after one round of duplication, that would be what you would expect. But at the point of duplication, you have a lot of things that are probably redundant, because you didn't need 2 copies before, and now you've got 2 copies. So it's very likely that some of these redundant copies would be lost. Some of them might be kept. If there's a benefit to having 2 copies, you might keep 2. And then over time, you might expect the different copies to become different from one another. So you end up with this sort of pattern where you've lost a few, you've kept doubles of some, but they still maintain this overall pattern of spatial organization. Now, if this undergoes a duplication event, remember the hypothesis is that there were 2 rounds of genome duplication in the vertebrate lineage. Now, these could be many thousands of generations apart. What you would end up with is this sort of thing after the second duplication event. Now, we've taken this and we've doubled it. Then we would also have another round, or another going forward from that duplication point, where some of these copies might be lost and some of these copies might pick up new functions. But they would still maintain this overall pattern. So what scientists realized was the way to test this hypothesis is to look in present-day vertebrates to see if the various gene copies, so these would be copies that are very similar to one another but don't necessarily do the same function anymore. The evidence is to look at these copies in spatial relationship to one another to see if there's evidence that these copies we see in vertebrate genomes now look like they are part of 4 sets of spatially organized genes in that particular pattern. The question then becomes, well, which genes do you look at? Because there's also evidence that evolution can produce new genes over time as well. So they wanted to be sure that they were looking at the right subset of genes. So what they did is they compared the genome of vertebrates with tunicates, and they picked out every gene that is present in these tetrapods and fish, and any gene that was also found in the tunicate genome. Those genes were most likely inherited from a common ancestral population before these different species went their separate ways. So by only looking at genes that are shared between all 3 species, we're looking at genes that would have been around for this potential duplication event. So when you look specifically at those genes in the human genome, this is what you find. So these are different human chromosomes. So human chromosome 10 and 2, 17 and 12. This is just one example of one subset of these genes that they looked at. The lines that connect the genes are just showing you where the 2 gene copies are in the human genome, or the 4 gene copies in this case. We've got 4, these are examples where there are 4 genes that are remaining, and you can see where they are spatially organized with one another. So look at this. We've got on human chromosome 17, we've got all of these genes in a spatial pattern that we also mostly see in part of human chromosome 2, a little bit on 10. Here's a big chunk of 10 that's also seen here. But you end up with this pattern where they do have what appears to be this overall structure that corresponds to this type of prediction. Now, if you look at genes that aren't shared with tunicates, we see this sort of pattern instead. So these are the cases where you have genes that are duplicated, but they're not thought to be part of that original set because these genes are not found in tunicates. They're something that cropped up after they went their separate ways. And here what we see is this sort of pattern where the gene copies are right next door to each other on chromosomes. So this looks to be a case where you have a gene and then it's duplicated, but it's just a duplication of one gene or a small amount of DNA that sits side by side in the genome, as opposed to this sort of pattern, which would be what results from whole genome duplication events. So all of that to say is that the human genome and the genome of any other tetrapod or fish appears to be not only a doubling of an ancestral genome, but 2 doublings of an ancestral genome. And here's the point. These proteins, these genes, don't have the same function anymore. These 4 copies, for example, have picked up different functions over time. So this is an example of new information coming into the system. So we have not only the ability for evolution to remove things, we also have the ability for evolution to produce things. So one other thing that Christians are very concerned about and very curious about in the present day is not only do we have an evolutionary history, the evidence that I've shown you is convincing to scientists that we do have an evolutionary history. One of the other things that Christians are often concerned about is whether or not we come from a population or whether or not we come from an individual ancestral couple in the past. So the whole question of, are Adam and Eve the sole genetic progenitors of the entire human race, or do we come from a population? Now, evolutionarily speaking, evolution happens to populations. It would be extremely uncommon for a new mammalian species to come on the scene simply through one single breeding pair starting a new species. Evolution seems to be this process where you have average characteristics within populations that are shifting over time. So for an evolutionary biologist, if you say humans evolved, they would think, yeah, okay, and they did so as a population. It would be pretty unusual if they didn't. We do have some examples of mammals on the planet that do seem to have come from very small populations. Cheetahs would be one example, and Tasmanian devils would be another example. From a conservation biology point of view, it's actually very challenging when you have species that have really small founding populations because they have such limited genetic diversity that it makes them susceptible to diseases and whatnot, and they're all uniformly susceptible. There's actually, in the example of Tasmanian devils, there's a transmissible facial tumor that's infecting Tasmanian devils right now. So it's a cancer of the mouth that when they bite another Tasmanian devil, the cells from their tumor get put into the other animal that they've bitten. Now, normally in most populations, that wouldn't be a problem because the recipient organism would recognize those cells as foreign and the immune system would fight them off. But because Tasmanian devils have such a low amount of genetic diversity, that transmission can take place and those cells aren't recognized as foreign because of the genetic lack of genetic diversity in those populations. As another example, you can have tissue transplants between any Tasmanian devil, between any cheetah, pretty much as well. And again, they're not recognized as foreign. Now, that's not what characterizes our lineage. We know that in order to do a tissue transplant, you need to have extensive screening to find a genetic match. And that is something that gives you a handle on the sort of genetic diversity that our population has. So it seems like, based on those lines of evidence, that at no time in our evolutionary history has our population been below about 10,000 individuals. There's lots of different independent ways to estimate those different population sizes based on current genetic diversity, and all methods that we've applied to date so far agree with this general number of about 10,000 individuals. So what that means is biologists or Christians are now beginning to have to wrestle with that particular issue theologically as well. But we can go back to our original discussion of two lines of evidence, or two sort of books of the book of nature and the book of Scripture, and begin to ask that question, we're now living in this interesting time where science is calling us to return to the text and to have that discussion. I sometimes say to my students, you get to live in an exciting time of history because the church is actually going through one of these issues at the present time. Right? We didn't get to live through the Copernicus thing where the church was wrestling through these particular issues, but you get to live in this time. And they said, well, I don't want to live through this time. I want it to be all nicely, I don't want to have to deal with it. And I said, it's not a bug, it's a feature. The fact that science can call us back to Scripture to make sure that we're reading Scripture appropriately for within its context and its setting, that's a good thing. Would you really, I asked them, I said, is it a good thing that the church shifted away from a geocentric understanding of the Scriptures? And they would say, well, yeah, of course, because it obviously didn't mean that. And when you look at it in the actual setting and context, you find out that that's not really what's being talked about. And I say, it was science that prompted us to go back to that. So science, in that sense, helped us correct 1,600 years of theology. I said, well, what do you think would be the situation if the church today maintained that, geocentrism was true? And they said, well, that would be a huge barrier to evangelism, right? Because you'd have to tell people if they want to accept Jesus that they have to ignore what science is telling them. And I say, yeah, and we're in that sort of situation now, but you get to live through this one, and maybe you get to be part of the conversation. I say, well, that's difficult. I don't like that. Well, it's where we are, and it's part of the natural process of how the church deals with these issues over time. Okay, let's land the plane. I've said that converging lines of evidence support the hypothesis that life on Earth is the product of an evolutionary creation. It seems like God chose the evolutionary process as the way for bringing biodiversity, including our own species, into being. I actually happen to think that's a fantastic way to design a biosphere, and I'll even use that word design, right? If God used an evolutionary process, that means that the biosphere is going to have species that are integrated correctly with one another. They're going to interact with one another correctly. And as that biosphere might shift over time in its characteristics, those species are going to be able to shift to those changing conditions as well. That's actually a very interesting way to design organisms in my mind. So if God designed life as an imperfectly replicating information generation loss system, such that the long process of speciation would bring about the biodiversity that he intended. So you can actually think of this, I sometimes tell my students, you're conditioned to think about evolution versus design. Maybe think about it along these lines. Maybe evolution is the design. Maybe that's the intended way that God wanted to bring about biodiversity. And last point, as Christians, we accept both that God works through what we perceive as natural mechanisms as well as through supernatural means. Both are forms of divine action, and both require the ordaining and sustaining action of God. So Christians have long accepted that God works through what we call miraculous means and also through what we perceive of as natural means. The challenge often is the case for Christians when something that we're used to thinking about in the miraculous category might get bumped over to what we consider the natural category. But that doesn't mean it's being bumped away from or outside of what God is doing. It's simply moving the category from what we consider miraculous to God's ordaining and sustaining natural features. And if you think that that puts it outside of what God is doing, then science is an inherently atheistic activity, right? Because anything we discover about the natural world we would say, oh, that means it's one less thing for God to do. But that hasn't been the Christian conviction over our history. The Christian conviction over our history has been that God is the author of both nature, and at times we see God acting miraculously within nature. So I would return to that particular diagram to discuss it. Okay, with that, I'll put up these as resources. I think John Walton's book, The Lost World of Genesis 1, is a great way to sort of start thinking about Genesis in its original context. If you're interested more about evolution as a scientific theory, I wrote a great big long series for BioLogos over the last couple of years that you could look at.